Good day and welcome to today's lesson. In our previous lesson, we looked at diversity of living things, that's classification, the first part of it, that is the part 1 of it. For today, we shall be looking at the second part, the part 2 of diversity of living things, that is the classification. And we were given some assignment to try at home, and I believe that you all did that. You can refer to your notes for answers. Alright, so our main focus for today will be kingdom prokaryote. Like we mentioned earlier, there are 5 kingdoms and we're going to delve deeper into these kingdoms. So we are going to start with kingdom prokaryote. And now one must note that the main organisms here in this kingdom are bacteria. Let us now look at the main objectives for today's lesson. And by the end of this lesson, you should be able to 1. State the main characteristics of the organisms in the kingdom prokaryote. 2. You have to explain the ways by which bacteria are classified. 3. You should be able to distinguish between gram-positive and gram-negative bacteria. And then lastly, you should be able to state the economic importance of bacteria. Let us now look at the main characteristics or the main features of organisms in the kingdom prokaryote. The word prokaryon is a 2-part name. That's a Greek word, and the word pro means before. The word karyon means nucleus in Greek, and so we put these two together. We call it before nucleus. When one says before nucleus, what do we mean by that? Now this is a nucleus that's lacks a membrane, a nucleus that doesn't have a membrane bound in it, or nuclear material without a membrane bound in it. So the first feature, really the main feature or the main characteristic of these organisms is that they lack a membrane bound nucleus. It means that they have nuclear material, alright, but they don't have a membrane surrounding the nuclear material. Then 2, they also lack membrane bound organelles. It means that their organelles are also lacking membrane. So they have ribosomes, alright, they have other organelles, but the organelles that they have do not have membrane surrounding them. Number 3 is that they have a cell wall that is made up of peptidoglycan. When I say peptidoglycan, it is a combination of proteins and polysaccharide. So from the word peptidoglycan, proteins combined with what? Polysaccharide. Now, one must note that peptidoglycan can also be called or be known as murine. So one must note that as well. So in your shot now are some images of bacteria or organisms in the kingdom prokaryote or the prokaryotic cell structure. So for the first one you can see here, now this is a bacterial structure. You can see that it has the ribosomes over here, and then they are not membrane bound. You can see that. Then of course you can see the nuclear material around this side. It is not membrane bound. The nucleus is there anyway, but there is no membrane surrounding the nuclear material. So let's take note of that one. Over here too is another thing, the same one. You could see this is the nuclear material that you can see, the nucleus, and it is not membrane bound. There is no membrane surrounding the nuclear material. So that is how come it is a prokaryotic cell. So let's take critical note of this one. Now, let's look at how bacteria are being classified. Now, there are several ways by which bacteria are being classified. We shall look at a few, about 4 of the ways by which they are being classified. The first one I'm going to look at is they are being classified based on their cell structure or form. Their cell structure or form. Then 2, based on the nature of their cell wall. Then 3, based on their oxygen requirements. And then 4, based on where they live or where they inhabit. So we shall be looking at, we shall be looking deeper into these ways in our subsequent slides. We shall now look at how they are classified based on their cell structure or their form. Now, based on their form or their cell structure, we put them into 3 main groups or 3 main categories. That is their shape, their structure, how they look like. The first particular one we're going to look at is the bacilli or the bacilli. Singularly, we call it bacillus. Now, these are rod-like or cylindrical shaped bacteria. They look like rod. They look like rod. And normally, they cause disease such as anthrax, typhoid fever, etc. When we say bacillus, bacillus simply means a rod, a little rod, little rod. So let's take note of that one. Then we have the cocci or the coccus. That is the singular form of the cocci, the coccus. Now these ones are spherical shaped bacteria, or they are round shaped bacteria. And the word coccus is also a Greek word which means berry. You see how berry looks like, round like that. So that is the coccus, and they look spherical or they look round. And they also cause disease such as gonorrhea, like the gonococcus. They also cause disease such as the boil, sore throat, piles, pneumonia, etc. So this is that one as well. Then the third particular one I'm going to look at is the spirilla or spirillus. Now this was taken from a Greek word called spirillum, which means coil. Now these bacteria are spiral shaped bacteria, and then they also cause diseases such as syphilis, etc. So these are the three main categories, or the three main forms within which we classify or we group bacteria, that is based on their cell structure or their form. Now one must note that, now you see in several books you see that they have another particular form called the vibrio, but you have to note that the vibrio is not a different form. It is under the rod-like bacteria, that is the bacilli. It is just a bent rod which forms a comma shape. So it's just a bent rod. It's also a rod-like one, but then it's just a bent rod. So you have to know that one. And it is the particular bacteria that causes cholera, the Vibrio cholerae. I think, remember, you've heard that one before. Yeah, so that is it. So let's note that the Vibrio is another rod-like bacteria, but just a bent rod that forms a comma shape. Now I'm going to look at the second way by which they classify the bacteria, or they group the bacteria. Now this is based on the nature of their cell wall. Now what the cell wall is being made of, or how the cell wall is like. So that's the other way that they group bacteria. Now based on these particular one, they group them into two main groups. That is, one, the gram-positive bacteria, and then two, the gram-negative bacteria. So we shall be looking deeper into these two bacteria. Now let's look at the gram-positive bacteria. Now these are bacteria that are able to retain the gram stain or the dye after a staining process. Now realize that normally before we study these bacteria, some of them, because of their nature, they look very light. So we need to stain them before we can study them under a microscope. Now the gram-positive bacteria, for them, after we have stained them, they retain the stain. So it means that they are able to absorb the stain, and then the stain stays on them after the staining process. Now this is because of one thing. Now remember earlier that we said the bacteria are having a cell made up of peptidoglycan. Now for the gram-positive bacteria, they have a very thick peptidoglycan layer, and this enables them to be able to stain, or to be able to retain the stain, or to hold the stain after a staining process. So let's take critical note of that one. So we call them the gram-positive bacteria. They are able to retain the stain after a staining process. Alright, so here is a typical envelope of a gram-positive bacteria. You can see that this is the envelope of the gram-positive bacteria. This is actually the cell membrane of it, and then it is indicating the cell wall over here. You can see this is the peptidoglycan layer. As you can see in your shot, this is a very thick one, a very thick peptidoglycan layer, and this is the reason why it is able to retain the gram stain after the staining process, because of the thick layer it's having here at the peptidoglycan wall. So take note of that. Now let's look at the gram-negative bacteria. Now if you know the gram-positive bacteria, then you should know the gram-negative bacteria. Now these ones, they have a very thin peptidoglycan layer, and because of this, they are not able to retain the gram stain or the dye after the staining process. So we call them gram-negative bacteria because they are unable to retain the stain after the staining process. So with this, you should be able to distinguish between the gram-positive and the gram-negative bacteria. For gram-positive, they have a very thick peptidoglycan layer, which make them able to retain the gram stain or the dye after the staining process. And for the gram negative, they have a thin layer of peptidoglycan, and this doesn't allow them to retain the stain after the staining process. Now in your shot is an envelope of the cell wall of the gram-negative bacteria. Now in your shot, you can see that the peptidoglycan layer here is thin as compared to that of the gram-positive bacteria and the gram-negative. So you see that the layer here is thin, and because of the thin nature, that is why it is not able to retain the gram stain after the staining process. Now let's look at the third way by which we can classify or categorize bacteria. Now this is based on their oxygen requirements. Now according to this one, we can have about 3 or 4 ways by which we can classify them based on their oxygen requirements. The first one I'm going to look at is the obligate aerobes. Now the word aerobe is not a new word to you. When you say aerobe, remember anytime you hear the word aerobe, aerobic respiration, it means that it has to do with oxygen, so don't forget this. So obligate aerobe. Now these ones are bacteria that require continuous oxygen supply for their growth and survival. It means that without oxygen supply, they can't grow, they can't survive. So they need continuous supply of oxygen before they can survive or grow. We call them the obligate aerobes. So let's take note of that one. Now let's look at the second group. We call them the obligate anaerobe. Anaerobe is not a new word to you, of course, because remember anaerobic respiration. Remember that one occurs in the absence of oxygen, so take note of this. The one is the obligate anaerobe. Now these ones are bacteria that do not require the presence of oxygen for their growth and survival. So these ones, they don't need oxygen supply for their growth and survival, so they can live without oxygen supply, and they can grow, they can survive. We call them obligate anaerobes. And the third one is what we call the facultative anaerobes. The facultative anaerobes. Now these ones are bacteria that can survive and grow with or without oxygen presence. So for these ones, they can survive whether there is oxygen or not, they can survive. So we call them facultative anaerobes. So let's take critical note of this one. Now let's get the fourth way by which we classify bacteria, or we group bacteria. Now this is based on where they live or where they inhabit. So according to this, we can classify or we can group them into 4 main categories. And the first and foremost is the thermophiles. Thermophile. Now the word thermo is not new to you. In physics, remember thermal energy. So anytime you hear the word thermo, it's related to heat or temperature. So let's take note of that one. So when we say thermophiles, now these ones are bacteria that inhabit areas with very high temperatures, very high temperatures, like sulfur or hot sulfur or boiling springs, or even in compost heaps where there is very high temperatures. So we call them thermophiles. So this tells you that sometimes you can cook your food, but there can be bacteria that can inhabit that hot food, because we have some that live in very higher temperatures, and so we call them thermophiles. So let's take note of that one. Now we also look at the second group under this particular category. We call them acidophiles. And you can see a very key word there, acidophiles. So these ones, they inhabit areas with very low pH, that is acidic areas, where areas very low pH, that's the acidic areas. We call them acidophiles. The word itself explains itself to you. So acidophiles, they are the bacteria that inhabit acidic areas or areas with very low pH. Remember that areas with low or pH that is very low is the acidic range. So take note of that one. Now the third group under this particular category is the halophiles. The halophiles. Now these ones, they inhabit areas with high salt concentrations, like the salt ponds. They inhabit high salt concentrations. So if you go to salt ponds, where you see that people harvest salts, realize that you can have bacteria inhabiting these areas. We call them the halophiles. They live in areas where there is higher salt concentration. We call them halophiles. So let's note that one. Then the last one is what we call the methanogens. Methanogens. Now these ones, they inhabit areas with anaerobic conditions. Remember, when we say methane gas is produced as a result of anaerobic respiration. So let's note it. So these particular organisms or bacteria, they inhabit areas with anaerobic conditions. So let's take note of that one. And one must note that the methanogens, normally sometimes they inhabit our intestines, and they can also cause flatulence. So let's take note of that one. They can cause flatulence. So let's not forget that one as well. Yeah, we should note this. It's very, very important. We should note that bacteria was first discovered by a scientist called Anton van Leeuwenhoek. He was a Dutch scientist, and he lived between the years of 1632 to 1723. Call him Anton van Leeuwenhoek. So he was the first person to discover bacteria. So let's take critical note of this. Alright, so let us now look at the economic importance of bacteria. Now the first one I'm going to look at is that they are used in treating sewage. When we say sewage, basically refer to a suspension of liquid or water and then solid waste. So basically referring to waste, suspension of water and then solid waste. That's the sewage. Remember that bacteria it's a decomposer, and of course, because of that characteristic, it can help in decomposing or causing a decay in this sewage, in a way helping to treat the sewage. And two, bacteria also causes diseases. They cause diseases. So we realized earlier we spoke of diseases such as cholera, gonorrhea, pneumonia, piles, boil, and so on and so forth. These are diseases that are caused by bacteria. And of course, bacteria can also be used to extract certain minerals. Yeah, that is another way, another economic importance of bacteria. Then the fourth one is that they cause fermentation during the production of cheese and then yogurt. So through anaerobic respiration, they can cause fermentation during the production of cheese and then yogurt. Then of course they are used in the production of some enzymes. So let's take note of that one as well. Then number 6 is that they are used in the production of drugs such as antibiotics. So let's take note of this one. Then of course lastly, they are used in the brewery industry. Because of their anaerobic activities. They are used in the brewery industries for alcoholic fermentation so as to produce alcohol for other alcoholic beverages. So these are some economic importance of bacteria. Thank you. Alright, so all too soon we have come to the end of today's lesson. I hope you enjoyed yourself. Let us look at a few questions that you have to take and then try. So this is your assignment. You try this. The first question is that you have to state 3 characteristics of organisms in the kingdom Prokaryote. Then 2, you have to distinguish between gram-positive and gram-negative bacteria. 3, you are to state and explain the 3 types of bacteria based on their oxygen requirements. Then 4, you have to state 4 economic importance of bacteria. Do have a nice day, and I'll see you in our next lesson. Bye bye.